The secret code of life. Billions of years before a single human heartbeat, before the oceans shimmered under sunlight, there was only darkness. Endless, empty, eternal space was quiet, except for the faint hum of creation itself. From that void, matter was born. Atoms collided, broke apart, and collided again, making patterns from chaos. And somewhere within that ancient dance of dust and fire, something miraculous began to stir the first hint of the whisper of life. But what is life really? Is it a pulse, a pattern, or something even deeper, a kind of code that allows the universe to recognize itself? For centuries, we have searched for that answer. From Aristotle's philosophy to Darwin's theory, every generation has tried to decode the invisible instructions that turned chemistry into consciousness. Yet one mystery endures. How did lifeless matter begin to live, and why does it keep evolving? To find that answer, we must travel back in time to an Earth that was anything but alive. 4.5 billion years ago, our planet was a violent newborn, storms of fire, oceans of acid, a sky of poison. It was chaos in its purest form. But in that chaos, something began to change. In the bubbling heat of a volcanic pool, simple molecules began to dance to a strange rhythm. They bonded, broke and bonded again, until one day a fragile structure emerged, a cell. It was microscopic, silent, and yet it carried within it the power to shape worlds. That single cell didn't know what it was. It had no purpose, no consciousness, but it could replicate, it could pass on information, it could remember. That memory, written not in words but in molecules, became the first code of life, the genetic code. The same code that billions of years later still beats inside you. For nearly 3 billion years, Earth belonged to single cells, tiny organisms invisible to the naked eye, but mighty in their persistence. They adapted, divided, conquered, and survived in every corner of the planet. But at some point, something extraordinary happened. A few of those cells began to work together. They stopped competing and started cooperating. And in that cooperation, life found a new way to exist. It learned to become more than one. That leap from single-celled to multicellular was the greatest revolution in biological history. It transformed oceans into ecosystems and paved the path to everything complex, everything living, everything conscious. But how could such a leap happen? How could a single cell decide to give up its freedom for unity? Scientists struggled with this question for decades until a quiet man in Michigan tried something extraordinary. In 1988, evolutionary biologist Richard Lenski began an experiment that would change science forever. He took a simple bacterium, Escherichia coli, and started to grow it. Every day, he would take a few cells from the previous day and place them into a new flask of nutrients. Then he would watch. The bacteria would divide again and again, day after day, year after year, generation after generation. Over tens of thousands of generations, the bacteria began to change. They became faster, stronger, smarter in their own microbial way. They learned to process food more efficiently. Some developed new metabolic abilities, traits that their ancestors never had. They were adapting before his eyes. After 75,000 generations, equivalent to millions of years of evolution in human terms, those tiny E. coli had become something new. Not a different species, but proof. Proof that evolution isn't just a theory written in dusty textbooks. It's a living process unfolding every second inside every cell on Earth. But Lenski's experiment, as groundbreaking as it was, left one mystery unsolved. His bacteria were still single-celled. They had evolved, yes, but they hadn't crossed that invisible line, the line of collective efforts between two cells. For that, the world would have to wait. 2 decades later, at Georgia Tech, evolutionary biologist William Ratcliffe decided to continue what Lenski began. Only this time, his subject wasn't bacteria, it was yeast, a single-celled organism that shares nearly 20% of its genes with humans. Yeast are eukaryotic, meaning their cells are more complex, more like ours. They were the perfect candidates to study how single cells might evolve into multicellular beings. Ratcliffe placed the yeast into nutrient-rich flasks and allowed them to grow generation after generation. Every day, he gently shook the flask and let them settle. The heavier clusters of yeast, the ones that stuck together, sank to the bottom. Those clusters were collected and grown again. This simple act of selection repeated daily for weeks. Then on the 60th day, something miraculous appeared. The yeast had formed clusters that looked like tiny snowflakes, delicate branching structures made of dozens, then hundreds of genetically identical cells. For the first time in human history, scientists had watched single-celled organisms evolve into multicellular one right before their eyes. But something unexpected happened. The clusters stopped growing beyond a certain size. They seemed to reach a limit as if nature itself was holding them back. So Ratcliffe and his team tried something bold. They recreated the conditions of Earth as it was 600 million years ago, a world with almost no oxygen. They placed the yeast into an oxygen-deprived environment and waited. And then it happened again, but this time faster. In just a few hundred days, the clusters grew larger than ever before. Thousands of cells bound together functioning as one. What took nature hundreds of millions of years, human hands had replicated in months. The transition from single cell to multicellular life had been reborn in a lab. But why did low oxygen trigger this transformation? The answer lay in survival. Without oxygen, yeast switched from aerobic respiration to fermentation. This new way of producing energy was inefficient and dangerous, exposing cells to stress and instability. The only way to survive was to cooperate to form protective clusters that shared resources and shielded each other from harm. In other words, life learned to evolve together because it had no other choice. The results were breathtaking. Under pressure, life didn't collapse. It united its walls, thickened its bonds, strengthened, and its collective intelligence emerged. For the first time, scientists could see the mechanics of evolution at work not as an abstract theory, but as a living truth. Decades earlier, another group of scientists had noticed something strange while studying bacterial infections. Inside the human mouth, certain bacteria didn't live alone. They formed sticky, complex structures called biofilms, slim shields that protected them from antibiotics and the immune system. Within those films, bacteria behaved differently. They communicated shared nutrients and even sacrificed themselves for the colony's survival. In other words, they acted like multicellular organisms. This was no coincidence. Biofilms might be the missing link between single-celled life and multicellular complexity, a bridge across billions of years. The bacteria that learned to form communities didn't just survive, they thrived. Cooperation wasn't the opposite of evolution, it was its secret engine. Everywhere scientists looked, they began to see the same pattern. From microbes in hot springs to fungi in forests, life evolved not by fighting, but by forming networks. It was never just survival of the fittest, it was survival of the connected. But one question still remained. If evolution could turn one cell into many, where did that first cell come from? How did lifeless matter awaken? How did the first code of life write itself? In a quiet laboratory at the University of Glasgow, chemist Lee Cronin and his team attempted to answer that question. They believed that life could emerge not from something magical, but from simple chemistry if the conditions were right. So they tried to build life from scratch. They mixed two water-based solutions, one containing carbon-based molecules and another containing metallic compounds like tungsten. When combined, something unexpected happened. The molecules organized themselves into tiny spheres membranes that behaved like primitive cells. These structures could store energy exchange, ions, and even self-repair. They weren't alive in the traditional sense, but they exhibited the basic properties of living systems. Cronin called them inorganic chemical cells. They were built not from carbon alone, but from the raw materials of the Earth itself. For the first time, scientists had created something that blurred the line between life and non-life matter, had learned to imitate biology. If these eggshells are anything like the first cells that formed billions of years ago, then the story of life is not a miracle. It is a process, a self-organizing system that begins wherever energy and matter collide under the right conditions. Life in this sense is the universe's way of learning about itself. The code of life was never hidden in a secret book. It was written in motion in the way atoms move, in the way molecules bond, in the way cells communicate. Evolution is not just the past of life. It is its language, a language spoken by everything that has ever lived. We often imagine evolution as a ladder, each rung leading to something higher, smarter, more advanced. But maybe it's not a ladder at all. Maybe it's a web infinite, interconnected, self-aware. And at its center is a truth so simple it feels divine. Life evolves not just to survive, but to understand. The secret code of life isn't written in genes or DNA alone. It's written in the desire of matter to organize, to connect, to endure. It's written in the curve of a spiral galaxy, in the pulse of your heart, in the flicker of a neuron firing in your brain. The same forces that built the stars also built you. And perhaps one day when we finally decode that language fully, when we truly understand how the universe gives birth to itself, we'll realize that we were never separate from it at all. Because the code we seek has always been alive inside us.